Hey everyone, welcome to the live class. We'll wait 1 more minute and then we'll start. All right, let's start. So, in the last biology class, I did cell theory. This was chapter number 1, by the way, for the IMAT syllabus, remember. So, I did chapter number 2, in fact. I didn't start chapter number 1 because chapter 1 is a bit confusing. So we started with chapter number 2. I finished cell theory. Cell theory was basically that our cells are made up of different organelles. I also did cell size. I did prokaryote and eukaryotic cells. Also the difference in animal and plant cells. I also explained viruses and bacteria. And today I'm going to focus on the structure and function of cell membrane and transport across the membrane. This is a very important topic and it's a big topic. So I hope we can finish this in today's class. So let's start with the cell membrane. Our organelles and cells are surrounded by something called the cell membrane, and all living cells are surrounded by this. In eukaryotes, remember, eukaryotes are surrounded by cell membranes. So this cell membrane controls how substance move in and out of cell and is responsible for many other properties of the cell as well. So the role of this cell membrane is that it controls movement of substances. What can enter and leave the cell is controlled by the cell membrane, and it's semi-permeable or partially permeable. What this means is that only some substances can enter. For example, water soluble, like uh, water soluble, or something that is like lipids, for example, are insoluble in water. So they cannot enter the cell because the cell is semi-permeable. So the membrane that surround the nucleus and other organelles are almost identical to the cell membrane. I said it earlier, all of the cells and organelles are surrounded by the same cell membrane, and the membrane is composed of these five compounds. So phospholipids, cholesterol, protein, glycolipid, which is basically carbohydrate and lipids combined together, and glycoproteins, which is basically carbohydrates and proteins combined together. Now, phospholipids will be discussed in more detail in chapter number 1. When I finish this chapter, I'll go to chapter number 1, and in that chapter, you will learn more about phospholipids. So this is the diagram of our cell membrane. This is also known as fluid mosaic model. You don't need to know what that name is, but you can see that there are different structures. So first there is the proteins in the cell membrane, and these circles that you see are called phospholipids. And phospholipids are the majority of the composition of our cell membrane. So most of the cell membrane is made up of phospholipids. And phospholipids, I'll get to that, but we also have this structure called the cholesterol. So let's get to phospholipids now. Phospholipid is made up of one glycerol molecule and two fatty acids. And there is one phosphate group. So one glycerol, two fatty acid, and one phosphate. Now, again, you'll learn more about the phospholipids in chapter number 2. But for now, you only need to know it's made up of glycerol. It has two fatty acids, one phosphate group, and the structure looks something like this. This is one phospholipid molecule. Now, this is what we call the head of the phospholipid, and the head is hydrophilic. What do I mean by hydrophilic? Hydrophilic means it's water loving. Heads love water, and they are charged, they are polar, so they can form hydrogen bonds with water because water is polar. So these heads love to form bonds with water because both the head and the water is polar. And the role of phospholipid is that it stabilizes the membrane. What do I mean by stabilize? I'll explain later on. But the tails, right, are hydrophobic. Hydrophobic meaning that it really doesn't like water. It doesn't form bonds with water. And so it's made up of fatty acid residue. So the fatty acid, the two fatty acids are present in these tails. And the tails are hydrophobic. That means it's insoluble and it's non-polar. So these tails, they act like oil. What do I mean by oil? They can't be mixed with oil. They don't dissolve in oil, and they are non-polar. So they won't form bonds with water. So they are non-polar. That means they will repel water. Now, they only small and lipid soluble molecules can diffuse into the cell membrane because the head is water soluble, the head is water loving, and the tails are hydrophobic. So only lipid soluble molecules and small molecules can diffuse. And what is the role of these tails? They maintain fluidity of membrane. So the head stabilizes the membrane and the tails help with the fluidity. So what is fluidity and stabilizing? Fluidity, right, is the movement of phospholipids and proteins in the membrane. So these phospholipids, right, these phospholipids, all of these are phospholipids. They like to move around, as well as these proteins, because the ions are present everywhere. They like to move around. So that is called fluidity. When phospholipids, proteins, cholesterol, they move around, it's called fluidity. And there are multiple factors that affect membrane fluidity. The number one factor is temperature. So the higher the temperature, the higher the kinetic energy would be, and the phospholipids, the proteins, all of that stuff would be have more kinetic energy. So they would be, this will cause more fluidity. So fluidity increases as temperature increases. Now, the next thing is the ratio of unsaturated to saturated fatty acid. If there are more unsaturated fatty acids in the tails, because remember the tails are made up of fatty acids, if there are more unsaturated fatty acids, then there would be more fluidity. But if there is more saturated fatty acids, there would be less fluidity. So unsaturated means fluidity is more. But if you want less fluidity, if you want to decrease the fluidity, then you would have to increase the saturated fatty acid. So if there is more saturated fatty acid, then there would be less fluidity. Now, why is this? This is because unsaturated fatty acids will cause kinks or repulsions between molecules, which results in more space. So when you have unsaturated fatty acids, there would be more space between these phospholipids. So these, this is one phospholipid, this is another, and there would be more space between these two if I have a greater number of unsaturated fatty acids. So unsaturated fatty acid causes kinks. Kinks mean that there is repulsion between molecules, between phospholipid molecules, which will result in more space between the phospholipids, and this will increase fluidity. So the third thing that increases fluidity is the length of the phospholipid tails. So the shorter the tail. Which tail? The tail of the phospholipids. These tails right here, which are hydrophobic. So the shorter the tail, the greater the fluidity. So the fluidity will be greater if the tails of the phospholipids are shorter. So longer the tails, less the fluidity, shorter the tails, greater the fluidity. And finally, the last thing which affects fluidity is cholesterol. Ignore this part for now. I'll explain cholesterol later on. But for now, you need to know that fluidity, when the fluidity is too high, meaning the phospholipid molecules are moving rapidly in the cell membrane, the length of the phospholipid tails would increase because you want to decrease the fluidity. So the tails increases, saturated fatty acid would increase, and cholesterol increases. What do I mean by this? Cholesterol would act as a regulator. So if you want to increase fluidity, you have to decrease cholesterol. So cholesterol is inversely proportional to fluidity. If cholesterol increases, fluidity would decrease. If cholesterol decreases, fluidity would increase. That was fluidity. Now, I did phospholipids. That was the first compound that makes up cell membrane. The second compound that makes up cell membrane is called cholesterol. Cholesterol, this is a cholesterol, and it's a small molecule. What is the role of cholesterol? It has a lot of roles, but cholesterol has hydrophobic tails and hydrophilic heads like phospholipids, and it fits between phospholipids. So when I have phospholipids, right? When I have phospholipids, cholesterol would be present in between these phospholipids. So this is a cholesterol molecule. It would be in between phospholipids. And it is not found in prokaryotic cells. It's only found in eukaryotic cells. What are eukaryotic cells? Our animal cells, plant cells, stuff like that, that are called eukaryotic cells. Now, what is the role of cholesterol? First role is that I told you earlier, it regulates fluidity of membrane. If cholesterol level is high, fluidity would be low or vice versa. And the second thing it does is it stabilizes the membrane. Stabilizing means that the membrane would not move more. It would stabilize the membrane. It would keep the membrane intact. And the third role of cholesterol is that it blocks passage of very small ions. So very small ions cannot pass through the cell membrane because of cholesterol molecules. And another reason is another role is that it waterproofs the membrane. Cholesterol is insoluble in water. So it doesn't like to form bonds with water. So it would waterproof the membrane. Now, another very important role of cholesterol is that it makes or synthesizes hormones. And not all hormones, only the steroid hormones. So cholesterol synthesizes steroid hormones. Remember that not all hormones are synthesized by the cholesterol, only steroid hormones are synthesized by cholesterol. So moving on to proteins, right? There are two types of protein in the cell membrane, the extrinsic or the peripheral proteins, which are the inner or outer surface of membrane. These proteins, right, the extrinsic proteins are outside the cell membrane or inside. Let me go to right here. So you can see there are these proteins, right? These proteins are present inside the cell membrane. So between phospholipids, these proteins which are present between phospholipids are called integral proteins. And the proteins that are outside or inside the cell membrane is called the extrinsic proteins. The extrinsic or the peripheral proteins. These proteins are present outside the cell membrane, not inside the cell membrane. So moving to membrane proteins. So I told you there is two types, the integral or extrinsic proteins. The extrinsic are outside or inside the membrane, whereas the integral proteins are integrated into the cell membrane. So they extend into the hydrophobic core or the hydrophobic regions of the phospholipid. So basically what this means is that it's present in between the phospholipids. So in between the phospholipids are called integral proteins. These proteins span across the membrane. They would be quite big. They can be inside the phospholipid, and they can also extend away from the phospholipids, right? So what are the role of membrane protein? There are multiple roles. The main role of membrane protein is they act as transport proteins. So transport proteins are only integral proteins. Remember that integral proteins, right? The proteins that are integrated into the phospholipids or are in between the phospholipids act as transport proteins, right? These proteins, the integral proteins, act as transport proteins, not the extrinsic one. The ones that are outside, they don't act as transport proteins. So what are transport proteins? They allow passage of ion, polar, large molecules through the membrane. There are two types of transport proteins. The channel proteins, right? The channel proteins are for facilitated diffusion. I'll explain what facilitated diffusion is later on. But for now, channel proteins for facilitated diffusion, and they are highly specific. What do I mean by highly specific? Only specific molecules can pass through these proteins. So I'll give you this right here is a transport protein. It's integrated into the phospholipid. You can see these are the phospholipids, and these are ions that are very specific, which are passing into the cell membrane. So the middle part, right? This part, right? The right here is hydrophobic. The middle part is hydrophobic. The head, bottom, and the hole through which the ions pass are hydrophilic. So this head right here, the hole that through which the ions pass, and the bottom, right? The bottom are hydrophilic. So only hydrophilic molecules that are water soluble can pass through the transport protein or the channel protein. And the second type of transport protein is called the carrier protein. This type of protein is used for facilitated diffusion and active transport. Again, I'll explain both what active transport and facilitated diffusion is. For now, remember that carrier proteins are also highly specific. It opens on one end and closes at the other end. What do I mean by this? Well, this is what carrier protein looks like. So this the protein enters from here. It's closed at the middle, but when the protein enters, it will change shape, and then it will look something like this. So this will close from the end, open, let the ion pass through, and then this side will open. So because of the changing of the shape of the carrier protein, it requires ATP, and active transport is what requires ATP. This reason, because the carrier protein will change shape, is the reason that carrier proteins require ATP. So example of carrier protein is a sodium potassium pump. You have to memorize this, that two potassium ions will be pumped in to the to the carrier protein or the cell membrane, and 3 sodium ions will move out of the cell membrane. And carrier proteins need energy for active transport because they constantly change their shape. I told you earlier, transport protein will change shape. So because of that, they need energy, and therefore they will need ATP. Now, what are the other role of membrane proteins? I told you earlier, they act as transport proteins. They also act as enzymes, right? And they act as receptors for cell signaling. I'll explain what receptors are when I get to cell signaling. But other than that, they act as cytoskeleton. They act as anchoring the cytoskeleton. Basically, they will maintain the cell shape, and they'll also link other cells. This is called cell-to-cell adhesion. So when two cells come together, they can these proteins can link these two cells. So ions can pass through these two cells. This is called cell-to-cell adhesion. Now, the fourth molecule of the cell membrane is the glycolipid. It is carbohydrate chains that are attached to lipids. So this is a phospholipid chain, right? And these are glycolipids. You only need to know the role of glycolipids, and that is that glycolipids, right? They would interact with water and stabilize membrane structure, able to form hydrogen bonds with water. Remember, only the carbohydrate part will form bonds with water because lipids can't form bonds with water. So the number one role is that they form bonds with water and they stabilize the membrane. Other than that, like proteins, they can also link other cells or cell-to-cell adhesion. They can also recognize cell. Glycolipids act as cell surface antigen. Antigens can detect foreign substances and trigger immune responses. So when a foreign substance like a virus enters the cell membrane, right? When this virus enters the cell membrane, the glycolipids can detect the cell membrane. They would act as antigens and they would trigger immune response. And basically, glycoproteins have the same roles. All of these roles also apply to glycoproteins. Right. So we're done with the composition of cell membrane. Remember the 5 different uh, compounds in the cell membrane: the proteins, glycolipids, the glycoproteins, phospholipids, and cholesterol. You have to memorize the roles. You have to memorize the structure of a few of these compounds. So now let's go to cell signaling. Cell signaling is when the cell detects and responds to a stimuli. And cell signaling allows cells to communicate with each other. This involves ligands. What is a ligand? A ligand is a specific chemical which acts as signaling molecules. So these ligands are secreted by cells or organs, and they act as signaling molecules. So what is the first step of cell signaling? The ligands are secreted from cells, right? Ligand is secreted from the cell. For example, adrenaline is secreted from the adrenal gland. Adrenaline is acting as a ligand here. And ligand acts as a stimulus. It's transported via the bloodstream. So any hormone, right? Adrenaline is a hormone. So it would be transported by the bloodstream, right? And this ligand transports via the bloodstream and would reach the cell surface receptor. So this is the bloodstream, right? And the ligand will reach the cell where it wants to do its response. So the cell has such cell surface receptors or protein receptors. I told you earlier, proteins act as cell surface receptors. So the ligand would bind to the cell surface receptor. The receptor are specific or complementary in shape to the ligand. So for example, this is the protein and this is the ligand. And the ligand and the protein are complementary in shape. they would fit in perfectly. So for adrenaline, right, targeted cell would be liver and muscle cell. So the liver and muscle cells would be targeted by adrenaline. They reach the adrenaline would attack liver and muscle cells. The shape of the receptor changes when ligand binds to the receptor. Now, what happens after the receptor, the ligand attaches to the receptor on the cell receptor? So receptors, right? The receptors on the cell membrane activate G protein. What is G protein? G protein is an enzyme or a protein which triggers the production of many secondary messengers by enzyme. So these messengers are known as secondary messengers, or simply this symbol right here. Secondary messengers trigger a chain of enzyme reactions, and this is catalyzed by phosphate or kinase. You learned this step 6 in the coming video classes. So enzymes, right, carry out specific response. So in a summary of cell signaling, right, this is cell signaling. So let me zoom in, right? So the ligand reaches the cell membrane. The cell membrane has protein receptor. The ligand attaches to the protein receptor. Protein receptor changes shape, activates G protein. G protein activates enzyme that makes secondary messenger, right? And secondary messenger activates enzymes that are specific to responses. So this part where secondary messengers activate enzymes for specific responses are called enzyme cascade. So this was cell signaling. And what is the role of cell signaling? There are multiple roles of cell signaling, and cell signaling would give you specific responses. What does cell signaling do? It gives you specific responses. What are examples of specific responses? So you want to increase transcription of a gene, cell signaling would happen, and there would be an increase in transcription. So I gave you an example of adrenaline. If your body wants adrenaline, then cell signaling would happen. The adrenaline gland would undergo cell signaling, and what this does is it makes you more active. So other examples of specific responses are opening of ions, change in cell shape, secretion of hormones, or secretion of different substances. Movement of ions are examples of specific responses. So that was specific responses and cell signaling. Now that we are done with cell signaling, I want to focus on transport of membrane. Now, there are 5 different ways through which ions are transported through the membrane. The first one is simple diffusion, osmosis, facilitated diffusion, active transport, and the last one is involved with vesicles. So let's start with simple diffusion first. What is simple diffusion? Simple diffusion is a few substances can diffuse directly through the lipid bilayer of our membrane. The only substances that can do this are lipid-soluble molecules, such as steroids, or very small molecules, such as water, oxygen, and carbon dioxide. Now, for these molecules, the membrane is no barrier at all, since lipid diffusion is obviously a passive diffusion process. So simple diffusion, you don't need energy, no energy is required, and simple diffusion is when molecules diffuse from a high concentration to a low concentration, from high concentration of molecules to low concentration of molecules. For example, let's say that this is our cell. this entire thing is our cell, and this is outside the cell. So this part is the outside of our cell. And you can see that we have a lack of oxygen in our cell. There is high concentration of oxygen outside the cell. So the oxygen would diffuse from a high concentration, meaning outside the cell, to low concentration or inside the cell. That was simple diffusion. What you need to remember is simple diffusion requires no energy and molecules diffuse from high concentration to low concentration. Osmosis is also very similar, but osmosis, instead of transport of ions, osmosis is the transport of water molecules. Again, it's from high water concentration to low water concentration. Osmosis is the diffusion of water across a membrane. It is in fact just like normal lipid diffusion. So this is example of osmosis. When you have low concentration of water, when there is less water molecules present outside the membrane, the water molecules would diffuse across the membrane so that you get more concentration of water molecules inside. So remember, water molecules will move from high water concentration to low water concentration. And what is facilitated diffusion? Facilitated diffusion is when you transport substances across a membrane through a transmembrane protein like channel proteins and carrier proteins. This is a passive diffusion process, meaning you don't need energy. And it is involved, and substances can only move down their concentration gradient. So from high concentration to low concentration. What's the difference between facilitated diffusion and simple diffusion? The main difference is that facilitated diffusion involves proteins. What are the two types? Channel proteins and carrier proteins. I explained both of these earlier. And active transport is only through the carrier protein. So carrier proteins undergo active transport. Active transport is the pumping of substances across a membrane by a transmembrane protein pump. This is called the carrier protein. And the main difference in active transport is that the protein binds a molecule of the substance to be transported on one side of the membrane. So the protein changes shape. Because it changes shape, this will require ATP or energy. And example of this I explained earlier was Na plus or K plus pump. So this transport protein is present in the cell membrane of animal cells and is abundant and important of all membrane pumps. So the Na plus K plus pump is a protein, and this protein would require energy because it changes shape, right? So the last method or mode of transport is vesicles. And the process described so far only applies to small molecules. But if you want to transport large molecules or large substances like proteins or nucleotides, you will transport these through membrane vesicles, right? And there are two types of transport involving vesicles, the endocytosis and the exocytosis. Endocytosis is the transport of material into a cell. Material are enclosed by a fold of the cell membrane, which then pinches shut to form a closed vesicle. This is how you form vesicles. You don't need to know this. What you need to know is endocytosis is when vesicles enter the cell, and exocytosis is when the vesicles, right, they move out of the cell. It is the exact reverse of endocytosis. And exocytosis happens in two main organelles, the Golgi body and the rough endoplasmic reticulum. Remember this part, rough endoplasmic reticulum and Golgi body, will produce vesicles and would undergo exocytosis. So exocytosis is when you secrete hormones into the cell or out of the cell. If you want to secrete hormones out of the cell or an organ, you would put the hormone in vesicles and secrete it out of the cell membrane through exocytosis. In summary, exocytosis is movement of material or vesicles out of the cell. Endocytosis is movement of vesicles into the cell. And exocytosis happens in rough endoplasmic reticulum and the Golgi body. And that is everything for this chapter. So we're done with this topic. So in the next class, I'll do cell cycle and cell division. We've completed the functions and the transport across the membrane. And let's do a question now. This is from 2021 biology. You have 1 minute to solve this question. It's easy. If you can't do it, I'll do it myself. Yeah, pretty easy. Which of the following is correct for cholesterol in humans? Sarah is saying A. A is it maintains fluidity of cell membrane. That is right. Cholesterol would maintain fluidity. If you want high fluidity, then then you would decrease the cholesterol molecule. If you want low fluidity, you would increase the cholesterol molecule. And B says that it is used to make all hormones. Only some part of the statement is true, that yes, it makes hormones, but not all hormones. It only makes steroid hormones. So B is definitely wrong. A is right. C says that it is a good source of energy because of the number of hydrogen bonds. No, this is not right. Cholesterol is not a good source of energy. So this is also wrong. C says that it forms hydrogen bonds with water, which stabilizes the cell membrane. That is incorrect because cholesterol is it does not form hydrogen bonds with water because it insulates the cell from water. So if you want to insulate the body from water, you would use cholesterol because cholesterol will not dissolve with water. And so D is wrong. And E says that it transports fatty acid around the body. This is obviously wrong because I never said it transports fatty acids. A is the right answer. So yeah, that is pretty much the class. I hope everyone learned something. See you tomorrow. Bye. Thank you for the class. You're welcome.